Hello, my name is Scott Ensel, and today I'll be presenting our work on the signature of consciousness in the transient dynamics of fMRI brain activity. So the idea of consciousness is a long-studied philosophical issue, but understanding how to detect conscious versus unconsciousness has clinically relevant applications. For example, people affected by locked-in syndrome, brain lesions, or head trauma will leave them in an unresponsive vegetative state while fully conscious. And current tests for this require difficult and active cognitive tasks. Now, previous studies have proposed that resting state fMRI can provide reliable biomarkers of the state of consciousness, and the results have shared hallmark brain changes independent from the nature of the unconscious state. Specifically, cortical long-range connections are disrupted in both space and time, and states are dominated by rigid configurations tied to their underlying anatomical connectivity. But by contrast, conscious wakefulness has been associated with a more flexible repertoire of functional brain configurations. So we hypothesize that these differences could be reflected in the spatial temporal reorganization of specific resting state networks and brain regions. Now, building on these results from previous studies, can we go a step further and not only discern conscious versus unconscious states, but also differing levels of consciousness using these fMRI biomarkers? So to try and answer this question, we used 143 resting state scans of non-human primates with differing anesthetics and levels of sedation. So indeed, the entirety of previous works use a sliding window technique hindering their conclusions. But here we overcome this limitation seen in those studies by deploying a novel method termed innovation-driven coactivation patterns, which captures transient brain activity. And the way this works is we take our noisy fMRI data, we can do some spatial temporal regularization to clean it, and then we can deconvolve this with our modeled response function and get an activity-inducing signal where we can take the derivative and get sparse innovations. From these, we can threshold this both spatially and temporally to get our significantly changing points. Now, we can cluster this together to get the most common spatial networks. And lastly, we can then back project these onto the original time courses and get overlapping temporal information. Now, from this entire bottom row of steps, we can extract quantitative data that's useful for our analysis. So if we start here with these thresholded areas, these transients represent large changes in fMRI activity, and we can plot the mean number of them for each condition. And what we notice here are significant differences in the awake condition versus the anesthetics, which highlight a reduced brain dynamic. And so to validate these results of these reduced dynamics, we individually clustered each condition, and the results are less spatial ICAPs, which validate this reduced brain dynamics. Now, when we cluster all the conditions together, we get out these 11 optimal clusters here. And so I'm going to go through and explain what this looks like. So in the top here, we have the secondary visual ICAP. In this bar graph here, it's a normalized distribution of how much each condition is contributing to the creation of this ICAP. And then here's the actual spatial ICAP that we have here. So now there's a couple ones that I want to specifically highlight, like the separation of the anterior DMN and the posterior DMN. And so you can only do this with our method, and later in the rest of the results, we'll see why this is important. So another couple to highlight are the anterior cerebellum and the posterior cerebellum, also separated. But one thing to notice here is that the posterior cerebellum is only found in the awake condition and not in the other anesthetized conditions. Now, we say that the posterior cerebellum is not in the other conditions because it's more motor action, and this absence has also been seen in other sleep studies. So I'll let you take a look at the rest of these for a second and kind of take them in and look at all the different ones that we have. Now, to capture the temporal dynamics of these ICAPs, we can perform that spatial temporal informed back projection here. And so when we look at the average durations, or how long these ICAPs would continuously last in a row, we can look at anesthetic versus awake. So awake down here, anesthetic up here, and we can see that this is significantly different for all of these durations. And if we were to break this down into each anesthetic versus awake, there are 4 ICAPs that we found to be the most significant across all conditions. And that's the secondary visual, the insula, the anterior DMN, and the prefrontal cortex. And they'll be more important later on. But to explore beyond the ICAP's individual temporal properties, we then evaluated the temporal overlap between ICAPs here. And so we computed the probability of having 1, 2, or more ICAPs occurring concurrently. And so ICAPs in the awake condition had a higher probability of occurring alone, whereas during anesthesia, they had a higher probability of co-occurring. So these results are consistent with the ICAP's individual temporal properties. And indeed, this longer average duration of ICAPs in the anesthetized condition resulted in increased co-occurrence of ICAPs over time. So to further explore these dynamic interactions across networks, we computed the temporal coupling of for every pair of ICAPs. So what we can see here is awake in the bottom corner of each one and the condition in the top right. So as expected, anesthetized conditions had stronger co-occurrence between pairs of ICAPs when compared to the awake condition. Some significant pairs were shared across anesthetics, whereas others were anesthetic specific. Specifically, we can look at ICAPs 1 and 2, which were commonly significant across all the conditions, ICAP 4, and ICAP 8. Now, all the ones that I just pointed out were the same ones that I talked about before that had those longer temporal durations. Now, this plot shows overall coupling, but ICAPs can be positively coupled or negatively coupled. And so of the ICAP pairs we found here that were significant, we then considered if pairs were more positively coupled or negatively coupled, you can see defined down here. Now, there's a couple things that I want to point out. The first is that larger lines means it's either more negative or more positive, where negative is blue and positive is red. And so ketamine here, out of all the conditions, had the largest ratio of negative couplings compared to the other conditions. So this was unique to ketamine. Next, the insula and the DMN are negatively coupled in most of the conditions, while the insula and the prefrontal cortex are positively coupled in most conditions. The next thing to look at is here we see a separation of the level of anesthesia with a secondary visual, and the anterior DMN are positive for lighter levels of anesthesia, and they're negative for deeper levels of sedation. The next thing and finally, one of the last things to look at is we see anesthetic specific couplings where the anterior DMN and the anterior cerebellum are negative for sevoflurane, and they're positive for propofol. So while generally ICAPs co-occurred more during unconsciousness for all anesthetics, different drugs, and more importantly, different levels of anesthesia induce pattern coupling and anti-coupling for specific ICAP pairs, so supporting the need to assess spatial temporal networks interactions to discern these alertness levels. And so since these results were all paired interactions between ICAPs, we expand this analysis to group interactions of the ICAPs. And in doing so, the ICAPs were grouped into two large clusters of both the sensory and the default mode network, where you can see the anterior DMN, posterior DMN versus sensory information here. So this was similar groupings to previous results in awake humans using the same ICAP method. You can see they have a sensory grouping and they also have a default mode grouping here. Now, to look at this in the other conditions, interestingly, this division was not preserved during the anesthetic conditions. Except for ketamine here, the anterior and the posterior DMN were split into two different clusters, further supporting the strong dynamical dissociation of the DMN into subnetworks, which can only be captured with the ICAP framework. So now unconsciousness is disrupting this hierarchical organization of brain dynamics. And these results are all these results I've just presented are all in non-human primates. So how do they compare to humans? And the next big question is, do these non-human primate fMRI results translate to humans? And the answer is yes. So we performed a summarized analysis of the spatial ICAPs found in 7 other ICAP studies done in humans and compared them to our spatial ICAPs. The difference we found came from the optimal number, where in humans it was 17, and monkeys, our study in monkeys, it was 11. But of the most common clusters found in these studies, we found a 1-to-1 match for every cluster we have, even the split DMN and cerebellum. And we believe this difference that we found is in this optimal numbers come from higher level cognitive networks such as the attention, anterior salience, and language and visual spatial, which were only found in the human studies. Yet the majority of our other networks were preserved, supporting the translational value of our results and justifying the animal model of our choice. So we've established that the functional brain networks in unconscious states have fewer dynamic fluctuations than conscious states, which results in longer temporal activations and more probable network co-activations. Importantly, pairwise and hierarchical network dynamics showed anesthetic-specific levels of consciousness traits. Additionally, previous works in sleep have reported a separation of the cerebellum and DMN, dissociating in the posterior and anterior parts upon reaching deep sleep, and in line with more recent studies that reported increased activation of the posterior DMN and lack of posterior cerebellum in deep sleep, suggesting a common neurophysiological role of the posterior DMN and posterior cerebellum in inducing unconsciousness. And lastly, these similarities open up the possibility that residual consciousness could be monitored through the dynamics of brain activity and tentatively suggest new biomarkers of conscious activity. So I'd like to thank my team. We have no disclosures, and thank you. Any questions, please contact me.